So in this video, we're going to be looking at an overview of photosynthesis, all right? And so just be aware that there are two main stages or reactions when we look at photosynthesis. The first one being a light-dependent reaction, sometimes it's called the photo reaction. It's where the photo and photosynthesis comes from. Also, it can be considered the light reaction because it's going to be requiring light to do. The other type of reaction is a light-independent reaction, also nicknamed a dark reaction. Now, don't get misled. A dark reaction does not mean that it has to happen only at night, right? It means that it's going to not require light in order to happen. And this involves the Calvin cycle. So the Calvin cycle is the reaction process that we consider to be the light-independent reaction of photosynthesis. All right, so let's look at an overview of the light reactions. Right, they're going to occur in the grana, or these thylakoid stacks in the chloroplast. All right, the chlorophyll is going to absorb solar energy and then convert it into chemical energy. So we can kind of draw like a flow chart, and you really can kind of sketch this in as an outline to add more information to. But we're going to start with light energy, and then that will be absorbed by chlorophyll, and that's going to energize electrons for us and move that along an electron transport chain. Ultimately, we'll be looking at a growing proton gradient. H plus ions will build up inside the thylakoid, and they will want to diffuse out. And when they do, they'll power or move through a protein channel that's attached to ATP synthase, which then can ultimately create ATP. All right, so that's one of the outcomes of the light reaction. All right, so in general, what we're looking to do is we're looking to split water, right, and release O2 or oxygen gas, and this is a kind of a byproduct, so it can be considered to be a waste of sorts. And then it's going to form this NADPH from NADP+. Right, this is an electron acceptor. It'll be carrying two high energy electrons with it, and these will be actually the final acceptor of electrons as they move through the electron transport chains. The dark reaction then is known as the Calvin cycle. We'll probably refer to it as Calvin cycle from here on out, and these are light-independent reactions. All right, so these will occur outside the thylakoids in the stroma. They'll use ATP for energy as well as NADPH for to reduce things, to help build sugars from the carbon that we supply, usually from carbon dioxide. So this carbon fixation process will happen. It's kind of like the main thing when it comes to Calvin cycle, and it's when we're going to, again, fix this carbon. We're going to fix it to a longer polymer chain of carbons and eventually form sugar molecules from that carbon dioxide. So how did we figure out that the oxygen that's released is actually created from the water and not the carbon dioxide that plants use? Right, so this goes back to an experiment that was carried out in like 1940 or around that year, and we had a oxygen-18 isotope. Right, so an isotope is simply the same element, but has more or fewer neutrons. In this case, it has two more neutrons than typical oxygen. So the idea was this: if we take this isotope and we put it with just carbon dioxide, right, and we can then measure the oxygen-18 isotope on these oxygen atoms that are molecules that are made. Then what we can do is run the experiment again and attach the heavy isotope to just the water, and then again see what the output is for the oxygen molecule. So this is what the results told us, and I'll move my pointer out of the way. Right, at the bottom I kind of highlighted that when we put the heavy isotope with water, we do find that the oxygen produced has that heavy isotope with it, and so that confirms that the oxygen that is produced comes from the splitting of water, not from the consumption or use of CO2. Pretty clever way to figure out that the source of oxygen being made is actually from the water molecules. Right, so at this point you should be able to make a table and look at where does these reactions occur. So the light dependent is going to occur in the thylakoids. Water, sunlight, NADP plus and ADP plus phosphate, an individual phosphate, will enter, and out will come the oxygen gas, the loaded ATP energy molecule, and NADPH. So essentially what happens is water split. It's going to result in the formation of oxygen gas, the release of a couple of high energy electrons, and also the release of H plus ions as well. The light independent reaction, or the Calvin cycle, is going to take place in the stroma. It's going to involve carbon dioxide, ATP, and NADPH. These are from the light reaction, and carbon dioxide is going to be pulled in from the atmosphere. Glucose will be the outcome, hopefully, some sort of sugar. It's usually a more simple sugar than this, and then this is built later on, but this is going to be our target sugar. And also, since we use energy, ADP and our NADP plus are also given off, and they'll be reused in the light reaction. So overall, what happens? Sugar is produced. All right, so this is simply an overview of the entire photosynthetic process on a very simple illustration. So we have, this is a chloroplast. It has the inner and outer membranes. This is the stroma, this area, outside. These are the thylakoid little stacks, and the entire stack would be a granum. Multiple stacks are grana. So the basic idea is in comes light, in comes water. The outcome will be energy-rich ATP, energy-rich NADPH, and then also oxygen will be released at the bottom there. And then these high energy molecules will go to the Calvin cycle, where we'll introduce carbon dioxide to them, and then ultimately they're going to produce a simple sugar, or G3P, right? So also the Calvin cycle will obviously give back the low energy forms of these energy carrying molecules, so they can be recharged in the light reaction. All right, so the light-dependent reaction in more detail. All right, so first we need to capture that solar power. So to do that, in the thylakoid membranes, they have these photosystems. And these photosystems are simply a bunch of pigment molecules. The primary pigment being chlorophyll A, some type of chlorophyll A, and then we have other various pigment molecules, accessory pigments, that form this like overall antenna, where it's going to be receiving this incoming radiation from the sun, and certain wavelengths will get absorbed, and that will excite electrons, which will be moved to the primary electron acceptor, right? And the first case is going to be directed towards a chlorophyll A of some type. So when light hits this photosystem, then this antenna will kind of direct all of that energy towards that primary electron acceptor in the photosystem, which would be chlorophyll A. All right, so we know that two different photosystems will actually work together to make this happen. All right, so the first one here is photosystem 2. And the story behind that is when trying to figure out how photosynthesis worked, photosystem 1 was actually discovered first. So they named it photosystem, and then they realized that there's actually electrons that come in to help this photosystem out from someplace else. And when they backtracked it, they found that there was another photosystem in play. So photosystem 2 gets its name because it was found second, right? But it is the first photosystem in the chain to start moving electrons along the way, right? So we'll have an incoming beam of light. That'll be absorbed, and they will also water's in place, and that will be split. It's called photolysis. Photo meaning light, and lysis meaning to cut or break. So we break this water. So we break this water molecule into oxygen gas and hydrogen ions, and the two electrons enter the electron transport chain. And as they move along this chain, they will also run some active transport for us by funneling in more H plus ions. We'll get into that in a second. Eventually, these will be used to help generate ATP. And then they'll enter the photosystem 1, which is the second photosystem in the series. And so we need more light to help recharge these electrons. They'll be then funneled towards the primary electron acceptor and moving along another transport chain until they eventually form NADPH, which is a molecule which carries 2 high energy electrons. Right, so that's pretty straightforward there. So let's look at this a step at a time here. All right, so the first photosystem in the membrane is photosystem 2. Remember, it says photosystem 2 because it was discovered second. It's going to use chlorophyll A. It's not important that you know P680. That's just icing on the cake, but in case people wanted more detail, I threw it in there anyway. So this is going to get electrons by splitting water, and it's going to also give off oxygen gas as a byproduct. Photosystem 2, the electron transport chain, will move electrons along it, and as it does, it's going to pump in H pluses from the stroma into the thylakoid. So we're going to start building up this positive charge. We have a diagram coming up of this. So we're moving things from low concentration to high concentration. So this is a form of active transport. So this flow of H plus then, it will want to get back outside. And as it does, the only place it can, because it has a positive charge, is to go through ATP synthase, the protein channel that is attached to ATP synthase. So this will ultimately power the creation of ATP. And they call this process or chemiosmosis. And it's simply looking at using the concentration gradient of some sort of ion or compound to cause it to diffuse through a membrane. Again, this one's using a channel, of course, to get through the membrane. So this ATP that's made is going to go to the Calvin cycle, ultimately. The second photosystem then in the membrane is photosystem 1. It's going to use a different chlorophyll A, but still, you can just remember it as chlorophyll A. And it's going to get these electrons from photosystems to electron transport chains. So they reach the primary acceptor, which is chlorophyll A here, this P680. It moves along the transport chain, also pumping in H plus ions, and then it enters photosystem 1. We energize it with some light, and then it's going to move into a second electron transport chain, and this one actually ends up making NADPH. So NADPH is the final electron acceptors at the end of the whole thing. All right, so electrons start out being moved in photosystem 2. They make that journey to photosystem 1, and then ultimately through another electron transport chain, they end up eventually at NADPH. All right, and then this is also going to go to the Calvin cycle. Okay, so I put several images in here when it comes to the production of ATP and NADPH. This one actually just focuses on ATP, but what I wanted to do was actually skip this one, and this one's a nice one as well, but I like this one, right? Because it does a nice job of showing you the pathways of everything, and so here we'll kind of break it down here. See, we have incoming light. This incoming light is going to strike photosystem 2, right? And in photosystem 2, we also have water, right? So these are the low water molecules here. They enter the photosystem 2, and this light is going to provide the energy to split it apart. It's going to be broken into oxygen gas, so O2, and also a bunch of protons. Now, to make oxygen gas, we need 2 oxygen atoms to make 1 molecule. That means we need 2 waters to make this happen. And so if we break 2 waters apart, that means there are 2 oxygen atoms, which will form into oxygen gas, and then we have 4 protons or 4 H pluses. The electrons then are going to be moved along the electron transport chain. And as they go through the chain, if you follow the green line here, you can see it goes through several different protein complexes that are built into the membrane. And they're simply, you don't need to know exactly what the names of these are, but what you need to know is that as it moves along its transport chain, it is pumping in H plus ions. So we're loading the thylakoid on this side, inside the thylakoid, we're loading it with H plus ions. Right? These 2 electrons need a little energy, so as they go through photosystem 1, more incoming light actually energizes that and will move it into, finally, this is like NADP reductase. We do not need to know the name of this, but what you need to know is that NADPH is the final electron acceptor when it comes to electrons being moved along the light reaction. All right, and then we have one of our 2 products made here. The second molecule, or second energy molecule that's made is ATP. So how does that happen? Well, remember, we built a bunch of this H plus ion. The concentration of it is really high now inside the thylakoid. So we did that through active transport by the movement of these electrons. And so naturally, H plus wants to leave. The problem is this semi-permeable membrane, this selectively permeable membrane, will not let ions go through. So there's actually a specialized protein channel which also has ATP synthase attached to it. So as these guys flow out, it'll actually index this little turbine here, little rotor, and this rotor will index in along with it ADP and a phosphate molecule, and they'll get combined into ATP along at the same time. H pluses flow out. So it's a very interesting idea, very interesting concept, and this is really cool the way it's structured. As far as like as these things kind of flow out, they kind of power this enzyme to form ATP. So that results in our 2 high energy molecules being produced. Now, a cool thing about this diagram, again, you can see that it has this like stepwise process here, and where it kind of ends at ATP synthase, and it has all the fancy names for all of the different proteins and enzymes that we have in place. The main thing, again, is going to be photosystem 2 with light and water, making oxygen, hydrogen plus ions, and electrons that will move through a transport chain to photosystem 1. From photosystem 1, they'll be re-energized with light energy and move along another transport chain to eventually forming NADPH. On the other branch of this story, we have the increasing concentration of H plus wanting to diffuse facilitated diffusion out through ATP synthase and helping with the formation, powering the formation of ATP.